आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़।
We bring you all a podcast series on STEM popularization, that is, science, technology, engineering, mathematics, and medicine, supported and catalyzed by the National Council for Science and Technology Communication, the Government of India. Program is produced by Media for Community Foundation.
Welcome to another episode of the podcast from the INDO-FRENCH Center for the Promotion of Advanced Research, CEFIPRA. Here, we highlight the groundbreaking collaborations between Indian and French scientists across diverse scientific fields. And now, let's listen from them.
My name is Subramanian. So I am a professor at the Department of Materials Engineering. I am also the, what I can say is joint chairman of the INDO-FRENCH Cell for Water Science. And I am also an associate faculty at the Interdisciplinary Center for Water Research at the Institute. And my association with France dates back to over, I would say, 25 years, when we established the INDO-FRENCH Center for Water Science with collaboration with the IRD of France. Now that collaboration has in a way triggered joint CEFIPRA projects being implemented. In the earlier CEFIPRA project, I was a co-investigator. Dr. Jean-Jacques Braun from IRD was the principal investigator from the French side, and Professor Natarajan from our department, who is now retired, was the Indian principal investigator. So I was in a way a co-investigator in that project, and that was dealing with the polar goldfields tailings, looking at gold from the tailings, so-called invisible gold, and that got out. And my second project is this project, which is dealing with chromite. Basically, as we all know, chromium is a very toxic element, and it exists in two oxidation states, that is +6 and +3. While the +3 state is actually a benign state, the +6 state is actually carcinogenic. So India has a major chromite mine at Sukinda in Orissa, and several mining companies are there. So we actually selected that site for our project. And my French collaborator is actually Professor Yann Subri from IPGP in Paris, and he's also with the University of Paris. So coming back to myself, my expertise is in the area of mineral processing. You can say in biometallurgy, surface and colloid chemistry. That's I would put as the broad area, and focusing on environmental technology. So this project is actually in a way a multidisciplinary project encompassing geology, soil science, metallurgy, mineral processing, biology, and of course geochemistry. So the French collaborator specializes in geochemistry. He also developed some isotopic techniques, and in this context for chromium 53, 52, two isotopes. So we were wanting to use these novel techniques to study the transport mechanisms of chromium after it is mined, and whatever residuals are there in terms of mine tailings, and the natural processes like monsoon which carry the chromium released from this overburden or tailings to the water courses. So we looked at the nearby water courses. It could be, I would say, the dumps of the mines, the water stagnation at the mine site, the effluents getting released from the mines to the water stream surrounding the mine and including ultimately to the river. So there is a Gamshala river which is actually finally joins the major river of river courses. So we were sampling. Of course, the target was to look at not only the solid waste but also look at the soil, look at the surface waters, look at the underground water in terms of borewell samples, and also look finally at some of the paddy fields in which rice is grown. So we wanted to see the retination of chromium as it passes through the entire streams as a release from the mine tailings. So this was our focus, and the Sukinda, as I mentioned to you, is in Orissa. So the Sukinda Valley was the site that we went to collect our samples, and of course we had the cooperation of the mining company there, and they were very kind enough to permit us to take samples, and also for looking at contrasting climate in terms of monsoon and the dry season. So we actually made some 3 field visits to collect our samples, and they were very cooperative in helping us to collect the samples. So that is in a brief about the objective of the project. In terms of what we have done, we, of course, as I told you, it was a multi-pronged approach. So we looked at initially the detailed metallurgical characterization of the tailings of the overburden. We used several techniques for characterization in terms of petromineralogy, in terms of X-ray diffraction, in terms of electron spectroscopy for chemical analysis, in terms of X-ray photoelectron spectroscopy, and all the scanning electron microscope, the transmission electron microscope, all the characterization tools, state-of-the-art characterization tools were used. Some of them available at the institute, and some that were done at France. The second part was actually looking at, as I told you, the mobility of chromium, attenuation of chromium to the water courses. So we collected water samples, that is not only the surface waters in and around the mines, also the rivers, finally where it joins the river, and also underground. We actually collected some borewell samples. These borewells are actually already located in the vicinity of the mines surrounding the mine, the villages there. So they were kind enough to let us to collect those samples. So we wanted to see what happens to the fate of chromium. As I mentioned to you, the 2 important things were the chromium trivalent and chromium hexavalent. And in France, the group that I am involved in, actually they have developed a very sophisticated technique, what is called as isotopic technique for looking at chromium speciation. And that is actually looking at the ratio of chromium 53 to 52 and see how it changes from a pristine water to contaminated water with chromium. As a result of, I also mentioned to you about the meteorological changes. That means the dry season, what happens, in the monsoon, what happens, and what are the signatures, both in terms of chromium 6 and chromium 3. So that was actually the samples collected were analyzed for both these species by standard techniques, and the isotopic studies were also done. So this is one important component of the project, which was handled by the collaborator in France, Professor Jan Simri and his group. And in terms of looking at collecting the water samples, we actually isolated some microorganisms which are present in the waters there. And we could we isolated a lot of microbes, but we selected 2 of them which had a potency to bring about the reduction of chromium 6 to chromium 3. So the two of them were actually Chromobacterium bolechium and Spingophysis macrogoltabida. So these two species we have identified. We have deposited the sequence in the gene bank. We used a 16XR RNA technique to actually do the genetic sequencing of these samples. Of course, the standard microbiological protocols, the other tests were also done. And we also looked at the bioremediation of the soil samples by treating the soil samples with these microorganisms. And we looked at what happens to the chromium 3 and chromium 6 independent experiments. And we used the kinetics, we used the pH as one of the modifying parameters. We also looked at the concentration of chromium. So these are very standard tests that we have done to see the potency of these microorganisms to take up chromium. And what was interesting is that if you start with the chromium 6 solution and you interact it with these microorganisms, at the end of the experiment, two processes take place. One is actually the biosorption, that is the chromium getting absorbed onto the bacteria. The other one is actually a bioreduction process. So bioreduction means in solution, we found chromium 3. We didn't start with chromium 3, but we started with chromium 6. But after interaction with the bacteria, we could detect chromium 3. So independent of that, we did surface analysis using X-ray photoelectron spectroscopy to confirm the presence of chromium 3 on the surface of these samples, both microbes as well as solid samples. Second important thing was a very unique study with the synchrotron facility in Soleil in France, where we did actually a profile as well as the speciation of chromium 3 and chromium 6. These were special scanning X-ray microscopy techniques in situ, where we actually load the sample along with the bacteria and see what happens to the species. So we actually identified chromium 3 there. To start with, we started with chromium 6, but after interaction, again, we could see that the reduction was taking place. And these experiments, surface analytical tools that were used, were further corroborated with some molecular, because we are going to study deeper into the mechanisms. So we used some molecular biological tools to see what is the reductase that is generated by the bacteria to bring about this reduction. So in a way, it was a comprehensive study looking at all aspects, both at the solid solution interface and in the bulk solution, and also doing the surface characterization of the species. So this gave us, I would say, very, very confirmatory evidence that chromium 6 is reduced to chromium 3 by a bioreduction process mediated by the bacteria. So that was one aspect of it which was done at the institute. The other one is that we wanted to extend the study to look at what happens to the role of these microorganisms, whether you could use them as sensors. So we developed a voltammetric technique, electrochemical measurement technique, voltammetry, where we use 3 electrode system, where the working electrode was actually these microorganisms impregnated on a carbon-based electrode. And of course you have the reference electrode and the inert electrode to complete the circuit. So we actually start with an electrolyte containing chromium, both chromium 3 and chromium 6. Then once you do the electrochemical measurements, you want to see what happens to the chromium get absorbed onto the actually the bacteria which is impregnated onto the carbon-based electrode. And then we wanted to see whether the process of deposition and subsequent stripping, cathodic stripping of the chromium, we developed a sort of analytical tool to look at the concentration of chromium as a function of, as I told you, concentration, how the concentration changes. And we wanted to know whether the spectroscopic measurements that we have, we can analyze for chromium-3 by UV visible spectroscopic technique using divinyl carbazide as a complex agent. We can also look at the total chromium using atomic absorption spectroscopy. So a third method was to look at this biosensor method, where you actually absorb and then desorb it. So you strip it and then you compute the concentration. So that has also worked well, and we have tested it with not only the standard samples but also the samples collected from the mines, the mine effluents. So there is a potential for commercialization of this biosensor. That is one aspect of it. The other thing, as I told you, was the important study of isotopic exchange, which we found out that the chromium-53 can be used as a marker to trace the mobility of chromium, because as I told you that if you take a very fresh tailing sample, it is enriched in chromium-6, but if you take an old tailing, due to natural processes of oxidation reduction in the mine site, it gets reduced to chromium-3. So the chromium-3 gets enriched in an old sample, which is actually in a way good because chromium-3, as I mentioned, is benign. So the natural processes of oxidation, both by chemical as well as by biological means, can bring about the reduction. Then if you come to a very fresh sample, it is enriched in chromium-6, so that is again an indicator that we should take some preventive measures for its disposal. So normally we use a reducing agent in the mine. They use ferrous sulfate to actually reduce the chromium-6, and when the water is let out in the effluents coming out of the plant, they have to dose it with a chemical like this. So that is one process they have adopted, but this is another. The biological process of remediation can also be used. So on the whole, you can say that we have developed a sort of holistic approach to look at the transport of the chromium by isotopic exchange processes. We have also looked at quantifying the dissolved chromium using various spectroscopic tools. We have also looked at biological processes of remediation as a solution to do the treatment for abatement of chromium, or certainly for reduction to the +3 state. So this, in a nutshell, I would say, are some of the research contributions of the project, which has the potential to be commercialized. So what is also important is we have had very fruitful discussions with the mine people. We have had discussions, we have shared our results with them, and the plant personnel are now aware that at some places the chromium concentrations are above the prescribed standards, so they have to take some steps to mitigate it. So now, in terms of some achievements, as I told you, the biosensor is one which has a potential for commercialization. The use of chromium-53 isotope as a marker to trace the history of the chromium as it gets dissolved from the tails is one important mechanistic study that can be adopted. In terms of benefits to the younger workforce that were involved, we had actually about one postdoc, 3 doctoral students, 2 master students working in this project, and at least they had an opportunity to visit France from here, and vice versa. From France they came here. They also accompanied us to the mine site. So the training of personnel is one important aspect. The second part is actually the sharing of information with the direct user industry, which will be actually very useful for them to in future adopt it as a mitigation strategy. So that, in a nutshell, is sort of highlighting what we have achieved in this project, and what are the future value addition that can be done by the plant personnel, and of course the young scientists working on this project can take it forward in terms of developing better technologies or other strategies to address this problem. CEFIBRA's what I would say the main objective to fund Indo-French programs is one of a very structured program. I would say it's possibly about 35 years old, and a bilateral cooperation can be significantly improved because of this. So we have had, I have been part of Indo-UK project, somewhat similar, but the call is not very frequent. It's a bit infrequent call. But what is important here is you have a very structured committee, council, which actually makes the calls, you review it. Then I think the whole process is very clear. You only have to find a French collaborator in working in a mutually competitive area, and then you can certainly write a good proposal to fund it. I did find that the advisory committee as well as the scientific committee, they are quite focused on what is expected from the project. The review process again was quite good. Of course, I was part of the midterm review in Delhi, and subsequently the next final review was held in France, so I didn't have to go there. In terms of the CEFIPRA structure as such, it's one of the best working structures for collaborative research between two countries.
This episode is brought to you with the content support from CEFIPRA and the technical support from the Sparrows team. We extend our heartfelt thanks to the Director of CEFIPRA, Professor Nitin Seth and Dr. Payal for their continuous guidance and support. Stay tuned for more episodes and thank you for joining.
आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ।